But Sierra, she got her bachelor's from North Carolina, Chapel Hill in biology and Spanish, and with a minor in marine science. So she's going to do today's in Spanish. We'll have an interpreter. She worked in the Florida Keys and the Cayman Islands, and as a marine science educator, conservation coordinator, and a scuba diving instructor before coming here to UVI. And here at UVI, she's worked with the Storm Strong program. You guys want to grab some seats? Or?
Oh, sorry, am I not there?
Okay.
Here.
And then she did some hurricane preparedness in schools throughout the territory. Yeah, come on in. Virginia, we have a lot of people coming in.
You can bring it back.
Okay, so while Sierra was at UVI, she did 2 internships. One with a did an internship with the Department of State involving climate and environmental diplomacy in the Pacific Island region. So when she started at UVI and started work on Redtail Parrotfish, all her lab mates sent notes of condolences because they're extremely hard to identify and they're really hard to catch, and there's nothing known about them. Yeah, but it's pretty amazing. We even had to contact an expert to look at their pelagic larval duration. So if you know anything about fish otoliths, they have little rings, like rings on the tree, which Sierra will describe, and nothing was known about this parrotfish or really any parrotfish in the Caribbean. So there's another Thesis project right there in the making for the pelagic larval duration, which is really important for understanding dispersal from spawning sites, how far larvae can actually disperse and settle into new habitats. And so she tackled every single challenge, learned lots of new skills, and produced a very nice thesis. So that's what she's going to be talking about today. Without further delay, she'll be talking about the juvenile life history of Redtail parrot.
Okay, cool. Wow, thanks, Rick, for that awesome introduction. So yeah, thank you guys all for being here. I am going to be talking about my thesis, which is titled Filling Juvenile Life History Gaps in a Data-Deficient Species, Redtail Parrotfish, Sparisoma, chrysopterum in St. Thomas, U.S. Virgin Islands. There we go. All right, so to get started, let's provide a little background of fishing throughout the U.S. Virgin Islands. So fishing is a part of the history, the culture, and the economy of the territory, and it provides a source of income, employment, and food source for people throughout the community. Now, while fishing throughout the territory is considered artisanal and small scale in nature, and most of the catch is geared towards local markets, it does contribute significantly to the economy, with fish landings averaging $7.4 million a year throughout the years 2005 to 2015. So small but mighty contributions. And in a survey of fishermen throughout the territory, they looked to see what fish were catching, and they found that 80% of fishers are targeting traditional reef fish species. And so while there are like technical advancements that's been made to allow fishers to fish offshore and for bigger pelagic species, a lot of the times they're still targeting these reef fish. And so when we refer to these reef fish targets, we're referring to our snappers, parrotfishes, triggerfishes, groupers, grunts, wrasses, and surgeon fishes. So you might be able to guess from the title which one we're going to be focusing on, and that is our lovely little red-tailed parrotfish. So this species is commercially and recreationally harvested throughout the territory, and it is in the parrotfish family, which is family Labridae, genus Sparisoma. And along with the other members of the parrotfish family, they are herbivores, and they've been classified in the functional group as browsers, meaning that they mostly like to consume macroalgae and turf algae throughout coral reef ecosystems. So one aspect of their reproductive biology is that they're considered sequential hermaphrodites that display protogeny. So what exactly does that mean? In simplified terms, that really just means that they start off their life as females, and then at some point, depending on environmental factors, they can make this transition to males. However, not all fish will do this. Some will remain females. So not every fish in the Sparisoma genus does that. Now, another unique characteristic about them is that they display these distinct color changes between their different phases of life. So to get a visual of that, we see our juvenile phase, which isn't very different from our initial phase. Coloration is pretty similar, but you see this drastic and complex color change that occurs between this initial and terminal stage that can be associated with that change in sex. Now, as I said in the beginning of this presentation, that we are looking at filling life history gaps. So when we talk about fish life history, what we're referring to is how a fish forages, how it ages, how it grows, reproduces, and where it's living throughout its life. And ultimately, the combination of all of these factors together, it is what is going to allow for fisheries to be managed sustainably. So it's important that we have all of these characteristics and traits. And this is a lot of information to obtain on a species. So there are lots of people out there doing this, but this study is specifically looking at the life history characteristics of juvenile habitat, as well as the relationship between habitat and growth during this juvenile phase. So as we begin to talk about juvenile fish habitat, many coral reef or fish that are associated with coral reefs when they're adults, they often use distinct juvenile habitats. And the reason for this is because this is thought to minimize mortality and optimize survival. And so these juvenile habitats are going to provide these ideal characteristics so that they can succeed in this and grow to adult populations. So some of these ideal characteristics are having a high abundance of food. So ideally, you need food to grow. So ideally, it has their preferred food sources. So turf algae, macroalgae for these species specifically. And ideally, a juvenile habitat is going to provide reduced predation levels. So while coral reef habitats might have larger fish, more predators, this juvenile habitat is going to have lower levels of that. Ooh. And our last point is that the juvenile habitat ideally is going to have more locations to shelter. So it's going to have a little bit added complexity potentially to allow fish to hide from any predators that are around. Now, another component of juvenile habitat is looking at how growth varies in these different juvenile habitats. And growth in juvenile stages has been described by two different hypotheses. The growth predation hypothesis and the stage duration hypothesis have kind of provided some background in that they've seen that if fish can grow faster, that they will be able to, they'll have higher chances of survival because one, they'll grow quickly out of the stages during their juvenile phase when they're most vulnerable. So they're getting out of these juvenile stages faster, and they're also developing these physical attributes that are allowing them to escape from predators or be large enough so that predators don't want to or are kind of deterred from getting them over the smaller individuals. So growth seems to be very important during their juvenile stages. However, it's also been documented that there are some trade-offs that occur with this growth, and that it's possible that a fish could have very high growth in one area, but maybe in this habitat where there is high growth, they might not have a lot of protection. So there could be this trade-off occurring, or vice versa, same thing in an area where there's not very high growth, there could be a lot of protection there. So then maybe they survive better in that area. So trade-offs to think about whenever we're thinking about this relationship. And so as a fish grows and progresses throughout its life, it is going to undergo what's called an ontogenetic habitat shift, or I should say it can undergo an ontogenetic habitat shift, which is basically just changing the habitat that it's in throughout its life stages. So in those earlier stages, it might need certain characteristics, but as it grows larger, it's going to want to transition to habitats that are able to better support them and provide those characteristics. So as we begin to look at how you would measure growth in a juvenile fish, maybe you're like me and had no idea what an otolith was before 2.5 years ago. And you thought maybe you measure growth by just taking the size of a fish. But there's actually a much better way to do it, which is through the use of otoliths, which are calcareous structures that are located in the ears of a fish. So this is basically a fish ear bone. And they are kind of similar to trees on a ring, or rings that occur annually on a tree if you were to cross section it open. But these rings occur daily throughout their juvenile life stages. And these can be really useful because these rings will continue despite environmental fluctuations and conditions. So while if you were to look at size, if a fish was maybe deprived of food for a couple days, the size would decrease of the fish and wouldn't necessarily provide an accurate estimation of growth. The otolith would continue depositing increments during that stage. So that's what makes it ideal for growth. So we have different parts of this otolith. The centrum is where the increments initially start to develop from. And then you have this larval period where increments are laid down while this fish in its larval stage is floating along in the water column. So you can be able to tell its larval duration from those increments. You have this period of metamorphosis where the increments start to look really wonky and different in all species. But with a metamorphosis, you can then be able to distinguish, once you know that point, then you can be able to distinguish where the post-settlement period is, which are marking the first days of a fish's life. So this is once the fish has gone through metamorphosis, develop all these physical attributes to be able to settle down into a specific habitat. So if we were to look at the growth rates, we're going to look at this based off of these increments, like I had said before, and we're going to be looking to see if you look at that distance, the white part in between two bars, that's what actually you're going to look at to get the growth. If there's a greater distance between them, then that's going to equal a higher growth rate, smaller distance, lower growth rate. So let's look at what we know so far about juvenile red-tailed parrotfish habitat from the literature. So we've seen that red-tailed parrotfish have been documented in seagrass and bare-bottom habitats in Biscayne Bay, Florida. We have seen in Curacao that they had a preference for seagrass and mangroves. However, when these two habitats were not present in the study, they were able to utilize other habitats. We also see that in Panama, they were found on seagrass beds of Thalasium testudinum that were on the fringe of coral reefs. And lastly, another study in Curacao found that bay biotopes, which were algal plains, seagrass, mangroves, channel, and like boulder, were all found and identified as potential nursery grounds. But with this, we really do not know a whole lot of these defining characteristics of where these juvenile fish are living and what habitats they really like to live in, and then how this growth differs between these habitats. So we have some data gaps that we are trying to fill for this species. And the reason that it's important to fill these data gaps is because for ecosystem-based management, which is kind of like the gold standard, ideally you would love to encompass all of these things, habitat identification and protection is really important, not just during the adult phases, but also throughout the juvenile phases of life to ensure adult populations are steady. So that is going to bring us into our first objective of what we looked at during this first part of the study. So we wanted to identify habitat distribution patterns in juvenile red-tailed parrotfish. So the questions that we asked, our first question was, how does the density and size distribution of juvenile red-tailed parrotfish vary among various habitat types? We also wanted to know how benthic characteristics are going to affect the density and size distribution of red-tailed parrotfish. And lastly, we wanted to know, are there any habitat shifts that could be occurring in these fish? So to do this, Let's go through our methods. So first off, we conducted underwater habitat surveys in 3 specific habitat types, which were rocky reef, hard bottom, and seagrass fringe. These habitats were chosen based off of initial preliminary roving fish surveys in which we had documented the highest abundance of juvenile fish in these 3 habitats, as well as from previous literature and then sightings whenever we were actually catching fish. So we conducted these surveys at 14 specific locations across St. Thomas that we clustered. We tried to cluster these 3 habitat types together across St. Thomas where we surveyed. And so that gave us 14 locations, but within each habitat type, we conducted 10 surveys. So the first component of these habitat surveys was to collect fish density. So we did this by belt transects, in which we laid down a 25 by 2 meter transect, and we recorded the abundance of the redtail parrotfish in 7 different size classes within that transect. This transect was standardized, the fish diver that did this, to 8 minutes, and then this was conducted along a randomly generated compass heading. So once we got down, it had a preset random direction that it would be laid out. Now, that was the first diver that went along conducting that. A second diver would come behind and look at the habitat characteristics. So those specific characteristics that we looked at were depth, topographic complexity, which that consisted of along a 15 by 2 meter portion, the first 15 by 2 meters of that transect. They would measure the highest hard bottom substrate within a 1 by 2 meter subplot of that. So what that ended up giving us was 15 measurements of complexity that were averaged together for that transect. We also looked at benthic substrate composition, which was ID'd as benthic substrate percent cover, in which we recorded the first 10 meters of the transect, and we analyzed that by coral point count methods, in which we overlaid these letters on the photos from the video and identified the substrate located beneath it. The substrates were identified according to the categories that you can see on the right-hand side. So sponge, coral, turf algae, dictyota species, macroalgae, seagrass, and non-living. So the last component of our methods was actually to take in fisheries independent data sources from the USVI to identify any potential ontogenetic habitat shifts that could be occurring. And so we took in data from the Territorial Coral Reef Monitoring Program that surveys coral reef habitats specifically. They survey in the same methods we did by belt transects and from the years 2003 through 2019. And we combined this coral reef data with my data from hard bottom and rocky reef to be able to identify some of these habitat shifts. And we did something similar with the National Coral Reef Monitoring Program that surveys 6 different habitats throughout the territory. And they have different survey methods, cylinder fish surveys. And we took the data from 2017, 2019, and 2021 from St. John, St. Thomas. And with that, we merged their habitat classifications, since they were more complex than ours, into just generally coral reef, hard bottom, and rocky reef. So let's get into the results of what we found whenever we did this. So to answer our first question, how does density and size distribution affect parrotfish among the habitats? We ran a Crystal-Wallace test to look at the differences in density between the habitat types. And we found that hard bottom had significantly higher density than rocky reef or seagrass. And when we looked at this by individual size classes, we ran a chi-squared test to look at differences between each individual, the density between each individual size class, to see if there was a difference between habitat type, the density between each habitat type. And whenever we ran this, you can see the asterisks. These are all the size classes that were significant. So all of them minus the largest, in which we didn't have enough data to actually run that test on it. So from this, we can see, and if you just visually look at this graph, we can see that hard bottom has the highest number of red-tailed parrotfish in these smallest size classes. So from 0 to 10, it's distinctly hard bottom that has the highest, followed by, again, that same pattern, rocky reef and seagrass. So we also wanted to check out these benthic characteristics. And when we did this, we ran a negative binomial regression. We did this because our data was over dispersed, had a lot of zeros. And so we looked at the two factors, complexity and depth, to see if it had a significant, if either of those characteristics were significant predictors of red-tailed parrotfish abundance or density. And so what we found is that complexity was a significant predictor. However, depth was not a significant predictor of density. So we looked a little bit closer, since complexity was significant. We wanted to look at this by size class and see what we saw. So we again ran that same negative binomial regression, except we ran this complexity by each individual size class. So when we look at the results of this, you can see highlighted in yellow that from 6 centimeters and on, all of those size classes, complexity was a significant predictor of density. However, in those two smallest size classes, that is not the case. What this could potentially indicate is that complexity in those very small size classes might not actually be that important, but overall, we do see that complexity is a significant predictor. Now, when we look at our benthic characteristics, we are looking at this percent cover data of each benthic substrate. And so with our three habitat types, rocky reef and hard bottom are way more similar than seagrass, which is classified by the presence of having seagrass, obviously. And so when we looked at the differences, we saw that hard bottom and rocky reef had the two substrates with the highest percent cover were Dictyota and turf algae. So we wanted to look specifically more at these two components of the benthos and see how they were potentially different between these two habitats. So we ran two separate t-tests. One t-test to look at the difference in percent cover of Dictyota between hard bottom and rocky reef, and then another one to look at the percent cover of turf algae between hard bottom and rocky reef. And what we found is that hard bottom had significantly higher percentage of Dictyota algae, whereas rocky reef had significantly higher percentage of turf algae. Now, we also, as you can see from those p-values, so we also wanted to look at our benthic characteristics, these substrates, a little bit more and see if any of them could potentially influence the density of red-tailed parrotfish. And so we ran, again, a negative binomial regression with all of the benthic substrate categories that we had identified to see if any of them potentially predicted red-tailed parrotfish density. And we did see that Dictyota was a significant predictor of density. So interesting that there is something going on with Dictyota, obviously in hard bottom habitat, and then across all habitats, we can see that it is increasing density. Now, when we wanted to look at to see if there were any habitat shifts, we are looking at all of those data sets kind of combined. So you can see in the very top graph, it's that TCRIMP data plus my survey data. The second graph below it is NCRIMP data set, and then the final graph is the roving fish survey data. And so when we look at our two smallest size classes, what we see is that their hard bottom, which is the pink color, has the highest number of red-tailed parrotfish being seen across all three of these data sets. And then as you look to the larger size classes, you see this almost like step-like progression of coral reef increasing, or of the number of red tails increasing in this coral reef habitat, which you can see in purple. So while we can't statistically compare any of these data sets because they are all kind of have different methods, different times, it is interesting to look at this trend and see that there does appear to be these habitat shifts. So for this first part of the objective, let's look at what we saw. So first off, we can see that red-tailed parrotfish are habitat generalists in nature. They were found in our surveys at all of the locations. It isn't like they are needing to have this one specific habitat as juveniles to make it to adulthood. We can see that they can utilize different, but it does seem that they have preference for specific habitats. And hard bottom appears to be one of those preferences that they have during that juvenile stage and the smallest size classes. We can also see that Dictyota algae appears to be an important habitat characteristic for these fish. As we had seen from the regression, that it did predict Dictyota presence predicted density of redtail. And one reason for this could be, if you look at our photo on the right-hand side, you can see that turf algae is very small and it doesn't provide anywhere for the fish to hide. It just basically covers any like hard bottom structure that isn't colonized. So there's not very much going on with turf algae. But if you look at Dictyota, what you can see is that it has shape, it has complexity, or you could call it like soft complexity, and that a fish would be able to, especially we're talking about fish that are really small, would easily be able to seek refuge in this Dictyota. And also red-tailed parrotfish are notoriously cryptic in nature. They can camouflage and hide very well into different things. And so I have personally witnessed a little juvenile red-tailed parrotfish really blending in with this. So that's also another interesting thing that we found. So if we also look at our complexity results, we see that overall complexity does increase density. But again, if we look back to that those two smallest size classes, they weren't affected by complexity. And then we kind of put that together with that this Dictyota could actually be providing that complexity that they need during the juvenile phase, not that hard bottom complexity that maybe they need as adults, which brings us into this next point of maybe why we're seeing these habitat shifts occurring. Because while our hard bottom had more Dictyota, had more of the soft complexity, smaller places to hide, that then as they shift to coral reef and could also be rocky reef, maybe intermediately, that they need that larger complexity to hide and seek refuge. And complexity has been documented multiple times in the literature that increased complexity is going to increase fish abundance. So that concludes the first objective. So let's talk about the second thing that we wanted to look at. So once we did all of these surveys, then we wanted to see how all of these different habitat types were potentially influencing the growth of red-tailed parrotfish. So we asked the question, is there a difference in growth rates of fish 0 to 5 centimeters total length between these three habitats that we had initially surveyed? So to do this portion, we collected fish from 3 different locations on the southwest side of St. Thomas. And within each location, we aim to collect 60 fish total, 20 fish in each habitat type, which ideally had a total of 60 fish in each habitat type. As you can see, we were just a little bit under that in a couple of the habitats, but we ended up having a total of 176 fish that we analyzed. Okay, so once we had our fish, we went through quite a process of, first we took measurements. So we measured the standard length, the total length, then we dissected it. And just to give a reference, these are like very small fish, 0 to 5 centimeters. So they were dissected under the microscope. And then we took out their we took out all their otoliths, but we were specifically interested in their lapular otoliths, which you can see are the smaller set of the 2 that are displayed there. Once we had those out, then we would mount them to a slide using super glue, and then they would be polished down with a really fine sandpaper. And so once we polish them down enough to be able to start seeing rings, then we would take some more measurements. And so this is where we actually began to look at those growth rings. So before we did that, we took these measurements, the larval radius, which again is from the centrum to the end of metamorphosis. The otolith radius is from the centrum to the end of the otolith. And then our post-settlement radius, which is from the first juvenile mark all the way to the end of the otolith. So these measurements were taken so that we could make sure that there was a strong relationship between otolith radius and then total length. So once we took our measurements, then we actually began looking at the increments. So this is a photo of one of the increments that we looked at. You can see in the blue again, that is our larval radius. And then we started taking the increments at that first mark, the first increment mark that is post-larval. So we made these increment marks for as long as we can, and then we ended up just we looked to see the difference again between these increments. Now we did this at the first 15 days post-settlement, but we also did this at the last 14 days prior to capture. So we took these increment mark measurements and growth rate mark measurements at two different parts. And so this in panel C, you can see our last 14-day increments prior to capture. So last 14 days of life. So whenever we look at the results of this, first, let's look and see what our fish size that we captured were. So from the histogram, you can see the number of fish that we collected by size, and the black bar represents the mean within each habitat. We ran an ANOVA on this, and we're able to see that from the graph on the right-hand side, that Rocky Reef had significantly higher, had a average total length that was significantly greater than both hard bottom and seagrass fringe. So this could be a potential effect of sampling bias. But we wanted to look at this and see, and we'll talk about what this could mean as we get into our other results. So this one specifically, where we looked at growth directly post-settlement. So again, this is looking at those first couple days of life. We looked at these increments in 5-day, we looked at the increments in 5-day increments over a course of 15 days. So a lot of increments going on. So what we found when we looked at the first 5 increments and averaged the growth, what we saw is that Rocky Reef had significantly higher growth rates than both hard bottom and seagrass fringe. And there was hard bottom was significantly greater than seagrass fringe. Now we continued to look at these results. We ran another ANOVA looking at days 1 through 10 post-settlement. And we saw this trend continued where Rocky Reef had significantly greater growth, followed by hard bottom and seagrass fringe. Once again, we ran another ANOVA to look at this full 15-day period of growth and see what was occurring. And we saw that the growth was again still highest in Rocky Reef, but there was no significant difference between hard bottom or seagrass fringe. So with these 3 ANOVAs, we can tell that growth is consistently higher in Rocky Reef throughout this first 15-day period of life. So there's obviously something going on in Rocky Reef that's making it important for fish that are initially settling in this habitat. Okay, so we also looked at the growth that was 14 days prior to capture. So these are the last increments, which you can see over here. And what we saw from this, whenever we ran an ANCOVA, which we ran an ANCOVA because we wanted to account for the fact that as fish get older, the distance between the increments gets smaller. So we wanted to account for that variable. We ran an ANCOVA, and what we saw is that there was no significant difference in growth during the last 14 days prior to capture. So let's talk about what these results could mean. Well, with growth being in the highest in Rocky Reef directly post-settlement, that's really significant that across this full 15 days, you can see this significant pattern that Rocky Reef is providing something that's making growth rates in this habitat higher. And during this post-settlement period of life, for juvenile period, it's when they're going to face the highest mortality rates of their life. So having higher growth specifically during that period where it's already really difficult for them is going to be really important and give them an advantage that could potentially increase their growth throughout life and increase their survivorship during this period. So but we saw, so you're like, okay, so if growth is higher during the first 15 days, why was it not higher or highest in the same Rocky Reef during that last 14 days prior to capture? Well, this reason is it could be that growth is really important during this initial post-settlement phase, as we kind of just talked about, but this growth might kind of level out as we kind of saw that trend happening and converging after days 1 through 15. You kind of saw the trend of there, while Rocky Reef was still higher, all of the numbers were kind of beginning to converge. So maybe after this first initial post-settlement period, the growth between the habitats isn't being affected as much. And again, this brings us back to our point that we had initially discussed in the beginning, that there could very well be these trade-offs that are occurring in these species. So while we saw from objective 2 in this part of the study that growth was highest in Rocky Reef, we, from the first part of the study, looking at the habitat surveys, we saw density was highest in hard bottom and juveniles. So this is like a prime example of how they might be settling in hard bottom and choosing hard bottom as their ideal habitat type as juveniles, because while it might not provide the highest growth, it might be providing these other forms of protection and refuge for them. Same thing, vice versa. Some fish might decide to settle in Rocky Reef and really go for that higher growth, but know that there might be that increased risk of predation. So trade-offs occurring that are complex. But let's tie both of these studies all together and figure out what this means overall. Well, the great news is that red-tailed parrotfish are habitat generalists. So it's a great thing for them in that they don't have to require one specific habitat. So for management purposes, it doesn't seem like any drastic measures need to be taken place for these guys. They can live in a variety of different habitats, but the important thing to note is that they utilize these nearshore non-coral reef habitats. And if we look at this map of habitats across St. Thomas, we can see that Rocky Reef, which is in red, really lines the whole coast of St. Thomas. And then it typically follows kind of a zonation of hard bottom behind that and seagrass fringe kind of scattered throughout. But all these habitats that they are documented in are nearshore non-coral reef habitats. So while we know we need to protect the coral reefs, it's also important in general to protect these nearshore non-coral reef habitats for juvenile fish. And especially for looking at this map again, if we think about where, if we're having coastal development or sedimentation and runoff, all of these factors that, we do have across the Virgin Islands, the habitat that's going to be most impacted is going to be Rocky Reef, as you can see, that really lines that coast and is found in very shallow areas. So just ensuring, especially since Rocky Reef was definitely an important component of survival and growth early on, important that these habitats are just noted for the future. So this brings us to, I'm going to bring us back to this little spinning wheel of all of the characteristics that are involved in a fish's life history that we talked about earlier. So we have growth, we have age, we have habitat, reproduction, diet. And this study is filling a piece of this puzzle, adding into this big wheel by contributing information about growth in the context of habitat and then juvenile habitat use overall, so that eventually we will be able to have this comprehensive understanding of what is going on in this fish's life throughout, all of its stages. So with that, here are my references. I'd like to thank my committee, funding sources, a lot of people in this room who helped me with field work. Thank you so much. Surveys wouldn't have got done without you. Taylor, thank you for all of your fish wrangling. Fish would not have gotten caught also without you. And yeah, my friends and family. So with that, I will take any questions. Okay, Kristen.
I have two questions. One, you said that you collected water depth information.
Mhm.
So I was curious if that was like a, you know, like a co-predictor of like, when you looked at the differences by habitat type, were those consistent across depths, or were the depths pretty uniform, so there wasn't a difference like that?
You're gonna need to repeat the questions just because we can't hear them online. You're gonna need to repeat the questions so that we know what you're answering.
Yeah, so she had asked if depth had been accounted for whenever we were looking at the differences in density between the habitat types. Yeah, so depth was we did look at depth, and I did run an ANOVA on the depth, and there wasn't a significant difference in between the depths. So between all of the different habitat types. So I don't think that depth necessarily was like significantly influencing the density.
Okay. And then my other question just had to do with like, I'm guessing that maybe this isn't enough, but like, how much do these little fish move? So like, in your 15-day, are you pretty confident that that's an integrated signal for, or even in the first 15 days or the last 15, that they're actually like, that's an integrated signal from that habitat?
Yeah, so she had asked about basically how much fish move, especially during that early stage of life. And yeah, that's a really good question. And whenever we ran the analyses on looking at the growth post-settlement, and whenever we captured the fish, we ran them under the assumption that the fish were captured in the same habitat that they settled in. So we operated under that assumption. Obviously, we don't know that for sure. And I'm not sure that there really is much literature to say like this is how much they move, especially for this species in that early life stage. So don't necessarily know like how much they could be moving to another habitat, but we did whenever we ran the analysis that we operated that whatever habitat we caught them from, we assumed that that was where they settled. Yeah, Nicole.
I had sort of the same question as Kristen, like going on with their habitat fidelity. Like some of your sites look like they had multiple like reef types in the same area. How far apart are those areas? Like where you, like on the map, it looks like some of your survey sites are like both rocky reef and seagrass. Like are those literally right next to each other, or how far apart are those?
So someone had asked like how far the habitat types that we surveyed are separated, and they are spatially adjacent. So they're all clustered together. So it would be, if you were to survey, you could go, which we did, we would survey like rocky reef, and then we would have to swim out to get to hard bottom, but they are located, like we wouldn't have to move the boat to get to the other habitat type. So they are located within this specific zonation and pattern. So all adjacent more so to each other, unless they were one of the sites that had just like one habitat type of interest. Allie?
Right. So you said that they're cryptic. Do you think that they are consistently cryptic across different size classes and across different like habitat complexity? And how do you think that might have influenced your observational abundance?
Okay, so there was a question on about them being cryptic and if we thought that that could potentially be influencing their the abundance in which they were recorded whenever we did these observations or our surveys. So I would say that since we did, the reason why we did a 25 by 2 meter transect instead of like the standard of what TCRIMP does, 25 by 4, was because we wanted to look specifically at the juvenile stage and like really get an understanding during that smaller stages of life. So I think that our surveys did a really good job of getting that juvenile stage. And I think because it was a smaller distance that anytime they were hiding or cryptic, we were able to document them within that. So I think it's more so like as you're swimming along, they have just like the tendency, you can see them in the water column, but they have a tendency, if they see you, then they will just kind of like merge and blend and hide. That's like their go-to. But despite that, I don't think that them being cryptic necessarily influenced the numbers so much because we did have that small area that we were surveying them for. I think was there anything else?
Do you think they were equally cryptic across the different sizes?
Across the different sizes? Let me think. I think
This is actually a really, really good question. So keep it in mind. This is a really, really good question.
okay. Perfect.
Yeah, so hold on to this. Don't let it go out of your head.
Okay. We're gonna ponder it for next little bit. Okay, do I think they are eagle? Let me see what I think. Wow, now I feel like the pressure's on for this one. Probably hitting the hard-hitting questions. I think that I noticed the bigger ones changing color and blending in more so, more than the smaller ones, I guess I would say. But all in all, I can't say that I really noticed like one size class being different than the others, like way more cryptic than other size classes. Yes, yeah, final answer. Nicole.
You were talking a little bit about there were different distributions in the Dictyota versus macroalgae versus turf dominated like habitats and substrates. Is anything known about their food preferences for either of those algae species, or like what they eat in general? And do you think if something is known that might also drive their distribution?
So Nicole was asking about the percent cover of specifically Dictyota and turf algae, and if it's known if that's their preferred habitat or preferred food source, and potentially how that could influence the distribution of the redtail parrotfish. So I would say yes, they're known to be browsers of specifically macroalgae and turf algae. So those are their preferred food sources, which yes is very important. And I think how they're distributed, especially since both of those habitats, while there wasn't a difference between hard bottom and rocky reef, both of those had the two highest abundances, and that's why I kind of focus more so on those. So yes, I definitely think that the presence of those algae types at a habitat could influence the abundance, especially with Dictyota, as we did see that it actually did predict density.
Other people on Zoom
yeah, I see one question on Zoom. Teresa.
Hi, I was just, this might be a very naive question because I'm not a fish person, but I have a memory of people doing experiments where they have a little newly settled larval fish or one that's just about to settle, and they have it in a little wine maze, and they have water coming from different habitats, and then they give the fish a choice. The fish can swim towards water from one habitat or water from another habitat. Do you think these fish would choose between your hard bottom and rocky reef habitats or seagrass in an experiment like that?
Oh, that's a really interesting question. I would love to run an experiment like that and see. Yeah, it's really interesting. Bring up a good point of like how they how they actually like choose a habitat. And from like the limited knowledge that I have on that component of it, it seems to be like a very complex process of like all of these factors that they're able to kind of like take into account before they settle into a habitat. So, you know, I don't know, but it would be really interesting to look at and see. I would guess that, yeah, at least Rocky Reef and hard bottom would have more fish that would want to settle in those as compared to our seagrass fringe is what I would guess. But that would be, yeah, a really interesting thing to look at.
Do you think it's a possibility that there's genes that, you know, some fish would be genetically ones that would go towards seagrass and others would go towards Rocky Reef?
I don't know. I'm not sure how like a genetic component plays into that and how they choose their habitat. So that's another really great question. I'm just not exactly sure how genes could potentially play into habitat selection.
I mean, theoretically, there could be a polymorphism in the population, and you know, those that like seagrass, you know, in some years would become more abundant, or if one habitat changes, how wonderful it is versus another, that might actually change the population genetically. Very interesting stuff.
Yeah, that would be really, really interesting to look at. Great question I had never really thought of before.
I can make a comment on that.
Oh, Rick knows a little bit. Rick is going to provide some insight.
It doesn't have to do with genetics, it has to do with growth. So I did work on bicolor dam bluefish. And the bicolor dam bluefish that grew faster while as a larvae and were larger at settlement went to coral reefs, whereas fish that grew slower and were smaller at settlement went into rubble habitat. And they eventually all moved to coral. So it's kind of like a trade-off you have here where there's more shelter and rubble than there is. So the largest fish went to the coral, so they had a size advantage at settlement. And made that, maybe, I don't know the decision, but that's significant difference.
Thank you.
That's that too.
We could just keep adding. So many questions.
Do another like few years.
So many gaps. So I'm doing my PhD. No, just kidding. Just kidding. Yeah, Rachel.
Amazing job, first of all. That was really, really good. So I am clearly not a fish person. So one of my questions, I have two. One of my questions is, how are you defining the juvenile stage with this fish? Like, you have a bunch of size classes, whatever. Are they all juveniles within the size classes? Or is it something you're introducing from the otoliths, which again is amazing that you were able to extract those from the tiny fish and extract them whole and not live them, because I had to try that once and I was very bad at it.
So if she's doing this, like the fish were actually that big, like sure, that's 5 centimeters, but she's taking some fish that are like freaking this big. I don't even know. Sorry.
So like shout out to yeah. How are you defining? what
Yeah,
juvenile
so
is?
these were definitely all the ones that we took the otoliths from were obviously juveniles, the 0 to 5 centimeter size class, and then beyond that, the prior to like reaching sexual maturity, obviously we kind of we had fish that were in some larger size classes, but that was just because we wanted to see all redtail parrotfish along this. If there were any, we were going to count them, but we were looking for the juveniles, and so that's more so defined as like size, like when it reaches sexual maturity, which Virginia can help me out on when that exact number is. But overall, like our focus was looking at that, like the earlier size classes, specifically. Yeah. So specific, well, I'm not sure if she heard the question, but yes.
And then question 2 is, how did you catch them?
How do we catch them?
Yeah.
So
It's the trade secret.
yeah. Yeah, they were caught with clear lionfish nets, in which we like scooped them up from the reef, or from whatever habitat we were in. So which is like literally 2 clear nets that we scooped together and collected them. So it was like really easy in the Dictyota, like they would be hiding and you could just on the Dictyota and there they were. So yeah.
Any more questions?
I don't think so.
Cast nets. They use cast nets too for some of the slave management.
No, not for any of these,
Not
for the
for any.
otoliths.
For the small ones, yeah.
Yeah.
Any more questions? No? Okay, thank you.